Hello everyone and welcome back to AP Bio with AP Playground. Today we're covering topic 4.6, Regulation of Cell Cycle. Today we're going to talk about the role of checkpoints in regulating the cell cycle. In 4.6 we discussed the cell cycle. Now we're going to talk about how it is actually controlled, as well as what happens if the regulation of the cell cycle is disrupted, which can result in cancer or apoptosis. And our suggested skill is argumentation 6E, predict the causes or effects of a change in or disruption to one or more components in a biological system. So the cell cycle control system consists of primarily three checkpoints that check whether certain conditions are met before the next stage can occur. First of all, there's the G1 checkpoint right after G1 that checks, is the DNA correct? Are there any mutations? Because we don't want to replicate and divide cells that have DNA with mutations. Can the cell keep growing and have enough energy to continue in the cycle? Essentially, is it going to be able to go through this whole cycle and then divide? And cells that don't pass this G1 checkpoint, the cell gets sent to G0. But generally, if a cell passes G1, then it'll tend to complete the cycle. Next, there's a G2 phase checkpoint before we enter mitosis that checks if the DNA has been replicated properly during synthesis. Because again, anything disrupting the DNA, then we can't divide because we don't want bad cells. And lastly, there's an M phase checkpoint. Have all the chromatids been attached to microtubules properly? Remember, in 4.6, we talked about PMAT in mitosis, where the chromatin will condense into chromosomes that are going to be attached to the mitotic spindle through microtubules, and they're going to line up at the metaphase plate, and then the mitotic spindle is going to pull all of the chromatids apart to create two nuclei. So if the chromatids haven't been attached to the microtubules properly, then they're not going to be pulled apart properly, and you might get a cell that ends up having an imbalance of chromosomes, with one is having more and the other one having less chromosomes, which can result in disorders and abnormalities and disruptions in function. Now, all of these checkpoints are controlled by cyclin-dependent kinases, which are only active when connected to cyclin proteins. Remember, a kinase is going to be an enzyme that carries a signal. So essentially, this signal is only going to be relayed when there's the presence of cyclin proteins. And these proteins are only present when the cell senses that it has the nutrients to grow, there's an abundance of space, all those things. And specific CDK complexes give the go-ahead at G1 and G2 checkpoints. Additionally, if certain growth factors are present, then other cells will be stimulated to divide. Essentially, do we have an abundance of nutrients? Because then other cells can start dividing and going through the cell cycle too. And there are two other checks that the cell makes in order to finish the cycle, and this is one, density-dependent inhibition, meaning if there are too many cells in one area of the body, then dividing needs to stop, or else there will be overcrowding and not enough resources for each cell. And also anchorage dependency. The cells must be anchored to something to divide, because we don't want dividing cells to end up in the wrong place of the organism. They have to be dividing in the certain place where they're going to be performing their function. They have to be anchored to divide. And what happens when something goes wrong with the checkpoints or the CDK complex, then there's a chance that we can get to a tumor, which is a mass of abnormal cells within normal tissue. And it's going to be composed of cancer cells, which have no limits on division. They're not density dependent. Even if the space is crowded, they will continue dividing and they will steal resources from other cells. They don't necessarily care about anchorage. And they'll divide even if there are DNA mutations and the checkpoint isn't passed. They'll find ways to bypass that checkpoint. And a tumor is a mass of abnormal cells within normal tissue. And if it remains at original site, then it's known as a benign tumor, while if it can invade and survive at new sites, it's a malignant tumor. Benign meaning good. Not that any kind of cancer is good, but it becomes more treatable when it remains at the original site. And metastasis is when cells separate from the malignant tumor and travel to other parts of the body, spreading the cancer. So it's so important that the cell cycle is regulated, because if there is abnormal cell growth, this can lead to tumors and cancer. So reviewing regulation of the cell cycle, it's important to remember that there are checkpoints that are testing to make sure whether conditions are met so the cell can continue through the rest of the cycle and divide safely, and this is largely dependent on the amount of cyclin present and the CDK complex. And lastly, cancer cells do exist and they generally do not follow any of the regulations of the cell cycle.